everyone, and welcome back to Nice Notes, Biology Edition. Today we're going to be breaking down cellular organelles, compartmentalization, and its mechanisms, a core topic in AP Biology that helps explain how cells survive, function, and interact with their environment. By the end of this episode, you should have a clear understanding of how cells are organized, how materials move across membranes, and why this matters to the real world. Let's start with basics. What is a cell? Cells vary in size, shape, and function, but all cells share the same foundational role. They are the most basic unit of life. Every cell has three main structural components: the cell membrane, cytoplasm, and genetic material, which in eukaryotic cells is enclosed in a nucleus. Inside the cytoplasm are structures called organelles, each designed to carry out specific tasks that keep the cell stable, efficient, and alive. This balance is known as homeostasis. Now, cells just don't exist randomly. There's a clear organizational structure in biology. Cells group together to form tissues, tissues form organs, organs work together in organ systems, and all of these systems together form a complete organism. This level of organization can exist across all living things, from single-celled organisms to massive multicellular organisms like blue whales. At each level, cells are the building blocks. Inside the cell, organization is taken even further through a process called compartmentalization. Compartmentalization means dividing the cell into specialized regions or organelles, each with its own environment and function. This allows the cell to carry out multiple processes at once without interference. For example, some reactions require acidic environments while others do not. Separating these reactions increases efficiency and precision. This brings us to organelles. An organelle is a specialized structure within the cell that performs a specific function. Similar to how organs function in the human body, some organelles are surrounded by membranes, while others are not. Membrane-bound organelles, like the nucleus and mitochondria, create controlled environments for chemical reactions. Non-membrane-bound organelles, like ribosomes and the cytoskeleton, still play essential roles, such as a protein synthesis and structural support. One of the most important structures in the cell is the plasma membrane, also known as the cell membrane. This membrane is found in all cells and acts as a boundary between the inside of the cell and the external environment. Structurally, it is made of a selectively permeable phospholipid bilayer, meaning it allows some substances to pass through while blocking others. Plasma membrane helps maintain homeostasis, protects the cell, and allows communication through membrane proteins, and controls the movement of nutrients and waste. In plant and bacterial cells, an additional cell wall outside of the membrane provides extra support and protection. A key property in the plasma membrane is membrane permeability, which describes how easily substances can cross the membrane. Permeability depends on factors like molecule size, charge, concentration, and the structure of the membrane itself. Small nonpolar molecules move easily, while larger not or charged molecules require assistance. You can think of membrane permeability as a coffee filter. Liquid passes through easily, but larger particles are blocked. This leads us to membrane transport, which is the movement of substances across the cell membrane. Because the membrane is selectively permeable, cells rely on two types of transport: passive transport and active transport. Passive transport does not require energy. Instead, substances move down their concentration gradient, from areas of high concentration to low concentration until equilibrium is reached. One type of passive transport is diffusion, where molecules move directly through the membrane. Another is facilitated diffusion, which uses transport proteins to help substances across the membrane. Third type is osmosis, the diffusion of water across the membrane. Water movement depends on the surrounding solution. Hypotonic solutions cause water to enter the cell. Hypertonic solutions cause water to leave, and isotonic solutions result in no net movement. In contrast, active transport requires energy in the form of ATP. Active transport moves substances against their concentration gradient from low concentration to high concentration. This process relies on protein pumps embedded in the membrane and is essential for maintaining ion balance, such as sodium and potassium levels in cells. Now let's shift to two main categories in cells: prokaryotic and eukaryotic cells. Prokaryotic cells are single-celled organisms that are considered the earliest forms of life on Earth. They include bacteria and archaea. Prokaryotes lack membrane-bound organelles and do not have a true nucleus. Instead, their DNA is found in a region of the cytoplasm called the nucleoid. These cells are small, simple in structure, and reproduce asexually through binary fission. Structurally, prokaryotic cells may contain a capsule for protection, a cell wall for shape, a cell membrane for transport, ribosomes for protein synthesis, flagella for movement, pill for attachment, plasmids containing extra DNA, and nucleoid region where the main chromosome is located. Eukaryotic cells, on the other hand, are much more complex. They have a true nucleus surrounded by the nuclear membrane and contain many membrane-bound organelles. Eukaryotic cells make up animals, plants, fungi, and protists, including humans. Because of compartmentalization, these cells can perform specialized functions effectively and efficiently and grow larger than prokaryotic cells. Eukaryotic cells contain structures such as endoplasmic reticulum, which helps synthesizes proteins and lipids, the Golgi apparatus, which modifies and packages these proteins, mitochondria, which produces ATP, lysosomes, which break down waste, and in plant cells, plastids like chloroplasts, who carry out photosynthesis. Before we wrap things up, let's connect this topic to current scientific research that builds directly on what we've discussed about membrane structure and transport. Recent studies in cell biology have focused on creating detailed maps of membrane transport pathways in human cells. Scientists are identifying and categorizing many transport proteins embedded in the cell membranes that move ions, nutrients, and even drugs into and out of the cell. This research goes far beyond simply labeling transport as passive or active. It shows how highly specific and regulated membrane transport really is, and how different transporters are responsible for moving different molecules across the membrane. This work is especially important in medicine because their understanding of these transport pathways helps researchers design drugs that can more effectively enter cells. Instead of forcing substances across the membrane, new treatments can be engineered to use the cell's own transport proteins, increasing efficiency and reducing side effects. This modern research directly applies to AP biology concepts of selectively permeable protein channels and the energy-dependent transport at a real-world scientific level. To wrap things up, let's connect this topic to antibiotics. Antibiotics work because of bacterial cell membranes differ from human cell membranes. Some antibiotics disrupt bacterial membrane permeability and interfere with transport proteins, causing essential processes to fail. Human cells are more unaffected because our membranes are structured differently. This allows doctors to treat infections effectively without damaging healthy human cells. Understanding the cell membrane structure and cellular organelles in transport isn't just about passing AP biology, it is about understanding how life functions at its most basic level, and that knowledge is applied in medicine and science every day. Thank you for listening.